am again. We have one more difficult and technical lesson before we go on to other more psychological considerations about the brain. So you have to tough it out for one more lesson. This is called synaptic transmission. And what you want to think about is this is one nerve cell, this is another nerve cell, and this is the point where they join, right? We know that happens a lot. When you feel something, your sensory nerve has to join to your the nerve of your brain to say, oh, this tastes good, or oh, this feels warm, or whatever sensation it is. It has to be communicated from one nerve cell to the next. And when you're thinking or remembering, there's intercommunication between interneurons. When you send a motor command, the neuron in your brain sends the command to the motor neuron. So there's always this junction, this meeting of one nerve cell meeting up with another. And the name of that junction is the synapse. Okay? The synapse is the junction between one nerve cell and the following cell. It could be a muscle cell, it could be a nerve cell. And amazingly enough, that synapse, which is shown here and here, if you were to zoom in and look at it, there's actually empty space. There's free space right there. And the name of that space is the synaptic cleft or the synaptic gap. Okay, I wanted to look back at a classic nerve cell just to remind you where we are in all of this. We're at the end of one nerve cell, so we're at these little things right here, which we I talked about them as frog toes because they bump out. And we're looking at this end point and how it joins or junctions with the next nerve cell or the next muscle cell. Here are different kinds of synapses. Here a nerve cell meets another nerve cell. Here a nerve cell meets a muscle cell. And here a nerve cell meets a gland cell, right, for the stimulation of some gland, such as a salivary gland or a digestive gland and so on. So there's all kinds of different synapses. Though this is a close-up of a nerve cell meeting a muscle, just to show you the little frog toes again. See those bumps? You don't need to pay attention to the labels. I just wanted you to zoom in and see it. Okay, so what happens at the synapse is super and cool. And by the way, it's super duper important for lots of psychological insights. Has a lot of bearing on psychological problems and psychoses. All right, so here we are again. This nerve cell is meeting that nerve cell. We're zooming in on this frog toe. Here it is. This is the frog toe, all big and huge and zoomed in. What exists at the frog toe? What exists at the frog toe's real name is axon terminal. What exists? Lots and lots of vesicles called synaptic vesicles. And they are full of a chemical. Here it's this bright orange ball called a neurotransmitter. Really important. Axon terminal full of vesicles. Remember, these vesicles are made of the same phospholipid bilayer as the cell membrane is. Full of vesicles containing neurotransmitter. Another picture of the same thing, right? And now we're going to start to go through it piece by piece. When the nerve impulse arrives, this is the nerve impulse arriving. See this? Nerve impulse traveling along nerve cell. It arrives. It activates this whole axon terminal. The first thing that happens is that calcium enters. So you don't have to understand this deeply. Just know there's all these ions at work here. And the calcium activates the synaptic vesicles, and they travel to the edge of the axon, and they release themselves. They release their contents through exocytosis. Okay, you'll see this when you watch the animated video. They release themselves through exocytosis. And remember what they're releasing? What's the chemical inside here? It's the neurotransmitter. And where does the neurotransmitter go? Well, now it's floating all around the synaptic gap. Let's look at it again. This is, let's relabel everything. The red thing represents the nerve impulse arriving, the action potential arriving. All of these are synaptic vesicles. What is the yellow thing they contain? The all-important neurotransmitter. When the action potential arrives, here they don't show it, but we know that calcium enters the axon terminal and activates these vesicles to release their contents. They release their contents, and now here it is, the neurotransmitter floating around the gap, the synaptic gap in this empty space. Here it doesn't really look like an empty space, but there's this empty space between the sending nerve cell and the receiving cell. Okay, let's go to this text here. When they go to the gap, the receiving cell, the cell which will be receiving the message from this nerve cell, has lots and lots of membrane proteins, all these purple things, and they're actually ion channels or ion gates. Can you see how this looks like an ion channel? That's where an ion travels through. These ion channels also have receptors. That's the blue thing. They have receptors. And guess what the receptors are made to do? To bind with the neurotransmitter. And notice what happens. When it binds with the neurotransmitter, the gate opens. So here we have a closed gate with no neurotransmitter bound to it. Here we have a neurotransmitter whose receptor has, an ion gate whose receptor has bound to the neurotransmitter, and therefore the gate has opened. So now all of these gates have opened up. Very often these gates are sodium gates, and what will flow through here is sodium, and the result will be that a new nerve impulse is initiated in the new cell. Okay, you might want to just pause and make sure you look at this to understand all the parts. This is even more of a zooming in on our synaptic vesicle. Here the neurotransmitter is triangular. Here the receptor site is this little triangular indentation. And this is a sodium gate which is closed. And when it bonds with the neurotransmitter, the sodium gate opens, and look what's happening. Sodium flows in. All right, so you can pause and look through that carefully. And let's look at what the overall result is. The overall result is that a nerve impulse, which was arriving in the old cell, in the sending cell, initiated the release of neurotransmitter. The neurotransmitter caused the sodium gates to be open. And because sodium is flowing into the new cell, what has happened? A nerve impulse has been transmitted to the new cell, to the new nerve cell or the muscle. Okay? So, all in all, the summary of everything that's happened is an arriving nerve impulse is transmitted across the synaptic gap and transmits the nerve impulse to the new cell. In fact, that's the reason for the name. The chemical is called neurotransmitter. It's transmitting a neural message, right? This is really technical. It will help a lot when you see it animated and watch the other videos. You should repeat this video and the other video twice to really start to grasp it. I think once you get it, it's kind of like riding a bike. Either you get it or you don't. So repeat it until you get it. A couple more details. Oh, this is just another image. Here the nerve impulse is blue. This is the sending cell. This is the receiving cell, right? The nerve transmitter is red. See how the gate was closed. Now the ion gates are open. The ion has flowed through, and the nerve impulse has been transmitted. Okay, so two more details. The first thing is neural stimulation would just keep going, right? You would just keep moving that muscle, or that thought would just keep stimulating the nerve cell over and over and over and over again. If something doesn't stop it. Well, what stops the nerve impulse? Two things. The neurotransmitter either disintegrates, breaks down, sometimes it diffuses away, or sometimes reuptake occurs. And this will be really important in future lessons. Reuptake just means that the sending cell regathers its neurotransmitter, takes it back in, and it's going to store it and reuse it. Reuptake is kind of like recycling. It says, I might need that neurotransmitter later on. So it gathers it up, and it'll be sure to use it again. This is the link to the YouTube video that you can watch on this. One more thing in my video that I want you to understand, and this is actually really cool, which is that there's two types of messages that can be sent. So this represents our new nerve cell. This represents old nerve cells. And notice, this is very realistic. The brain is not simple. It's not a linear sequence of one nerve cell leading to another, leading to another. It's a huge, complex interconnection. So look, how many nerve cells are synapsing, are meeting up with this nerve cell? 1, 2, 3, 4. 4 of them. And often it's many more. So these are two represented by 2 green ones and 2 orange ones. Okay. Now, actually what happens is that some nerve cells transmit an excitatory nerve impulse, or an excitatory neurotransmitter, which transmits a nerve impulse, and some an inhibitory one. And so I have a really simple but silly example. Let's pretend this is the nerve cell for crying, for your tear ducts to start making tears, and you're going to start crying. Okay? What could these orange ones, which are transmitting or exciting the nerve cell, be doing? They could be feelings of pain and sadness, right? Someone has just hurt your feelings. It's exciting this nerve impulse, and it's telling your nerve cell, yeah, stimulate those tear glands. I need to cry. I'm sad. Okay, so these are excitatory. What would the green ones be? The green ones would be your determination to get control of your emotions, or your determination to not embarrass yourself and not start crying. And they would be sending an inhibitory message, which would be saying, don't fire this neuron. Don't stimulate those tear ducts. I don't want to cry. Okay? So that's just kind of a colorful example to give you an idea how both of those might be firing at the same time, right? One stimulating the nerve and one inhibiting the nerve. So on a technical level, what does an excitatory neuron do? Well, we already looked at it. It's what we looked at before. It opens up sodium channels and it lets sodium flow in, because when sodium flows in, it stimulates a new nerve impulse. All right, so the excitatory ones are the ones we already talked about. What would the inhibitory ones be doing? How would they be restraining this nerve cell? Inhibitory neurons and the neurotransmitters they release make chlorine negative ion channels open. Why is that the case? Because if a nerve cell becomes more negative, it is very, very difficult to begin an action potential. So it's inhibiting, it's preventing an action potential. An action potential occurs when the negative interior of the cell becomes positive. And if you have negative ions flow in, it's further than ever from becoming positive. So it's inhibiting a nerve impulse. This is super complicated, so just skip this. You don't need to worry about that. So that's a very tricky but last thought about neurotransmitters. All right, like I said, I think this is challenging. I think you should watch the video I suggest, and there's plenty other videos on YouTube if you like. Listen to my video twice. Hopefully it will be clear. And email me with any questions. Maybe we can set up a Zoom call for questions. I think we'll probably need to do that soon. Enjoy your day. The weather looks nice, and enjoy your weekend. This assignment isn't due until Monday. Okay, take care, girls. Bye-bye.